Hey guys, it's Miss Carlson again. Today we're going to talk about Chapter 19, which covers bacteria and viruses. We're actually going to look at the similarities and differences between the two and identify which one is actually considered a living thing and which one is not. Now, make sure you have a good notebook sheet of paper or a Cornell note-taking template to take good notes, and we're going to go ahead and get started. And when a lot of people reference bacteria, sometimes we envision an organism that looks like this. We obviously know they're not green, have little tongues and whatnot, but they can be helpful or they can be harmful. Now, viruses, you may not have ever seen what a picture of a virus looks like, but they're very robotic and mechanical looking, as you see in a typical diagram here. Now, let's go ahead and start talking about bacteria. We have to remember that they are considered prokaryotes, which means they are unicellular, they do not have a nucleus, and they are separated into two different kingdoms. Now, there are some small differences between the Archaebacteria and the Eubacteria. The Archaebacteria live in extreme environments. They lack peptidoglycan in their cell walls, but their DNA sequences are very similar to that of a eukaryotic cell. Now, the Eubacteria are what we more commonly see. They can live almost anywhere, soil, humans, whatever, that's what we mean by anywhere, and they do contain peptidoglycan in their cell walls. Now, here's a diagram of typical organelles and parts of a bacteria. Now, here you have something called a pilus, or multiple pili, which are little hair-like structures that are sometimes used for locomotion. The flagellum is also something that is used for locomotion for the bacterial cell. And then you have some chromosomes, or nuclear DNA, again not enclosed in a nucleus because they're a prokaryotic cell. They have cytoplasm, cell membrane, cell wall, but that's about it. So not very complex cells. Now, bacteria are generally defined by their shape, the type of cell wall that they have, so again that peptidoglycan or the lack of it, and then the type of movement. So first, let's talk about the different shapes. There's bacilli are rod shaped, and you can see they kind of look like pieces of rice. Then you have the cocci are circular shaped, and they're generally in different kinds of forms. So just a circular bead-like shape, but they come in tetrad forms, or a group of four. They come in chains, as you see here with the streptococci type of bacteria. So many different formations. There's even something called grape-like clusters. And then you have your spirilla, or your corkscrew shaped, which are not of the more common types of bacteria. The bacilli and the cocci are the more common. Now, some bacteria, like I told you, are propelled by flagella. These are whip-like structures. Others can lash, snake, or spiral forward. Some examples of bacteria and what they actually cause or how they affect us is E. coli. They are a rod or a bacilli-shaped bacteria, and they usually give you the runs. Neisseria-Gonorrhea gives you the STD, gonorrhea. You have Streptococcus pyogenes, which causes strep throat, as well as a few other things depending on the strand. And then Treponema pallidum, it causes syphilis, which again is another STD. Now, these are all negative types of bacteria. There are positive bacterium out there that we will also get into. The overall breakdown is that bacteria are unicellular living organisms, okay? They are prokaryotic cells. They have all the 8 characteristics of living things. Now, viruses do not. They do not possess all the characteristics of living things. Most importantly, they cannot reproduce on their own. So they are not considered living. So bacteria, living, virus, not. Now, bacteria obtain energy in the following ways. They can carry out photosynthesis, and they are known as photoautotrophs, or they can obtain energy by taking in inorganic molecules, and those are known as photoheterotrophs. If unlimited space and food were available, a normal prokaryote can reproduce every 20 minutes, and then if left alone for 48 hours, it would actually reach a mass of roughly 4,000 times the mass of the Earth. Now, we know there are limiting factors out there, so that isn't necessarily a problem, but that just shows you how fast they can actually reproduce themselves. So let's start talking about viruses now. Like I said, they do not possess all the characteristics of living things, and one of the ways that makes them different from a bacterium is how they reproduce. Now, you don't have to write all of this down, but kind of put the gist of the differences and what they have that is the same. A bacterial cell will actually increase in size. It will have a double thick wall created down its midline, and then the cells will separate there, and then there are two new cells that function as a separate unit. So it's very similar to mitosis in some ways. Now, a virus actually requires a host cell. The virus will inject its genome or DNA into the host cell. It will then get copies made. The virus will assemble its parts. Once it's fully developed, it will actually explode into the host cell and destroy it. And this is actually something called a lytic cycle. Now, bacterium and viruses do share something called a lysogenic cycle, where this begins with a viral reproduction, then uses the bacteria reproduction to make more copies, and then goes back to the viral reproductive part. So a lot of information there, just kind of is FYI. Just note that the virus needs a host cell. The bacteria can reproduce on its own.
In a lysogenic cycle,
and this is a video that's going to show you how a virus reproduces.
And is replicated with it. Eventually, the virus enters a lytic cycle and kills the host cells. A bacteriophage attaches to a bacterial host cell by locking onto a specific receptor site on the surface of the host cell. The virus then injects its DNA into the host cell. Once inside the cell, the viral DNA inserts itself into a specific site in the chromosome of the host cell. Viral DNA is then called a provirus. During cell reproduction, the host cell copies the provirus genes along with its own DNA. The provirus is inactive at this time. When the cell divides, both the host genes and the provirus genes are passed on to the two daughter cells. At any time, a provirus may leave the host chromosome and enter a lytic cycle. When this happens, the host cell's DNA is broken down and new virus parts are produced and assembled. Eventually, the cell ruptures and new virus particles are released.
Okay, so that was a quick overview of the virus and both lytic and lysogenic cycles. There's also a diagram here to show you the difference between the two and how they are interconnected. Now, here is a electron micrograph of what a virus actually looks like. So you can see it does look very similar to that kind of robotic diagram that you were shown, but it's kind of nice to see what the realistic picture looks like as well. Now, I mentioned bacterium and how they reproduce and how it's kind of similar to mitosis, and so this is how bacteria reproduce in its process. It gets larger, they duplicate their chromosomes just like mitosis. The cells continue to grow, and then a wall will be placed in between the two cells where they would separate, and you would have your two new bacterium cells. So this is known as binary fission. Or again, the reproduction of a bacterium cell. Alright, so what is the difference between disease based by a virus versus one caused by a bacteria? So here's another important way to distinguish between the two. Diseases caused by a virus would include things like chickenpox, AIDS, and the common cold. Diseases caused by bacteria include strep throat, tuberculosis. In either case, we would call these pathogens. So viruses or bacterium that cause some sort of disease are known as a pathogen. Now, the main difference between the two of them is that a bacterial is its own single cell, and again can reproduce by themselves. The virus needs a single cell to attach itself in order to live. They are alike in the fact that they both disrupt the normal activity or homeostasis of the infected organism. Both can destroy cells. Bacterium can also release toxins harmful to the body. Now, there are different ways that we can fight bacteria and viruses that are dangerous to us. One of them is a vaccine, which is just a biological preparation that improves the immunity to a particular disease caused by a virus or bacterium. I will say vaccines are more commonly used for viruses, though. They usually contain an agent that resembles a disease-causing microorganism, which is often made from a weakened or killed form of that microbe. What ends up happening is that a vaccine will stimulate the body's immune system to recognize that agent and destroy it, so that later on, if you get infected by it, it will be taken care of by your body on its own. Now, bacterial diseases and infections are usually cured with antibiotics. And the word antibiotic comes from the Greek anti, meaning against, and the word bios, meaning life. So again, we talked about bacterium being its own life form, and antibiotics are also known as antibacterials. Now, like I said earlier, there are bad bacteria, but there are very good bacteria that play a helpful role in the ecosystems. They can make chemical compounds for many food products. They can aid in the preparation of vinegar, butter, and cheese, help in landfills by breaking down dead matter, and they're also nitrogen fixers. One example of that is rhizobium. It's a soil-based bacteria that attaches itself to the roots of plants. This is a bacteria, even though it needs a host plant. However, it cannot get the nitrogen itself. And here's a picture of rhizobium, which, do you remember what shaped bacteria this would be? It's a rice-shaped or a rod, and hopefully you guessed bacilli. If you did, you are correct. All right. There's another bacterium that's called Pseudomonas Fluorescer This. It's used in milk and dairy products to help them not spoil. And there's a picture of it. Again, another common rod-shaped or bacilli-type organism. And that is it. So make sure you go back and pause and play as needed. Don't overdo it. Just kind of take some brief notes on some of those longer parts. It's just more information for you to have. Not necessarily you need to know every little bit of that, but good to know. So I will see you guys next time.